From MicroTV, this is Office Hours for Wednesday, July 5th, 2023. I'm Vincent Racaniello, and welcome, or as Steph, one of our moderators, just wrote, greetings, people of the viruses. Now, what does that mean? Well, it could mean that you like viruses, or it could mean that you are viral, because we have a lot of viruses in us, and probably without them we wouldn't do so well. Anyway, welcome to your corner of the internet where you can talk about viruses or anything else. And I have for you tonight a Spike t-shirt, see? And it says, Vax Gratitude. I figured this would be a good one, an appropriate shirt to wear tonight. I ordered a couple more today. I really like this design, and the shirt's good. Remember, we did a fundraiser for MicroTV last year and raised quite a bit of money, so they hit a nerve, interestingly. So welcome, everyone. Let's see who's moderating tonight. Just about everyone. For a while, it was just moderators and nobody else. Steph is here. Andrew is here. Tom is here. Who else? I saw Frank. Haven't seen Frank in a while. Nice to have you back. And yes, we already saw Tom. So missing is Les. Maybe Les is somewhere here and has not said anything. That's always possible. And Barb UK is missing. Thank you for moderating again this evening. We might have a interesting live stream depending on who shows up. But first, a couple of pictures I wanted to show you. First of all, here's one from the recent meeting of the American Society for Virology, which was in Athens, Georgia. This was taken in the poster area. You can see these boards some of them have posters clipped on them. That's how people can present their work. They make a nice poster with figures and text, and they stand in front of it at a appointed hour, and they talk to people. And one of my favorite things to do, listen to this, what I like to do. I like to walk down the middle. And, I look at one, I look at the other, I look at the other, and I wait for someone to say, "Oh, can I show you my poster?" It happens very rarely. I want to see who is excited enough to overcome that barrier of being shy. It's hard to reach out to someone, especially some senior person who, and it's hard to do. So I wait for them. Of course, I also look at posters I'm interested in a priori, but I think it's very cool. And every row will have someone who reaches out, and I love it. I absolutely love it. Anyway, this group, by the way, is a group of virologists from Brazil. They're all Brazilians as far as I know. And there's, of course, Kathy Spindler and me here. And there's no Eurico, who is, the senior guy here from Brazil, but he's not here. He's not in the picture anyway. But this is great. I've seen many of these people before at ASVs. I've been to Brazil twice. I love it. There's great peoples there, and they are very friendly, possibly among the friendliest people on earth. Okay, that's Brazil. Then another one here. This is a picture commissioned by Barb Mack UK, and I have to make it smaller here so we can see everything. Okay, it's going to be next to me. All right. Now, this, of course, is the plaque assay princess, none other than Amy Rosenfeld. And so Barb Mack had this commissioned by an artist, and she gave it to Amy, and I understand Amy printed it out, and she is framed it and is going to hang it in her office, which is cool. But it has some defining features of Amy, and I'm sure you will recognize them. The shoes, the cashmere sweater, the lab coat, the little name tag with Amy, of course, the princess crown. and the plaque assay plate where viruses are coming out of it. And if we zoom in on that plate, maybe you can see, look at that, D68, enterovirus D68. Anyway, thanks for that, Barb Mack UK. I probably should have saved it to a time when both Barb Mack and Amy were here, but I couldn't wait. I think it's pretty cool. A little manga stuff, right? Okay. Now, many of you have seen, of course, the episode of Beyond the Noise that was just released this week. Vaccine misinformation with RFK Jr. It's gotten 19,000 views in just 2 days, which is pretty good. A lot of comments. And, of course, people want us to debate RFK, which is the whole point of the episode, is that you don't debate things that are facts, right? Like, yeah, all the vaccines have been placebo tested. That's not debatable. And if you want to define what a placebo is, you can't. No. You can have your own opinions, but you can't have your own facts. And placebos are not what RFK thinks they are. But anyway, there's no point in debating because, as Paul said, it will just get down to a shouting match saying, you're wrong, no, you're wrong, no, you're wrong, and that's useless. So what we're going to do is simply keep hammering at this. Now, next Friday, not this Friday coming up, but the 14th, Dan Wilson, will join the entire TWIV team, and we will go through this Rogan RFK Jr. podcast point by point and tell you why everything he says about vaccines is wrong. Of course, Rogan can't call him out because he doesn't know anything about vaccines. So that's the 14th. It'll drop on the weekend. I'm looking forward to that. That should be fun. That was Dan's idea. I think it's a great idea. So one thing that I don't get, and maybe you can explain this to me. Let me turn off this screen share for a moment. So over on Instagram, there's somebody who's being very nasty to me. So they don't agree with a lot of what I say, and that's fine. I don't mind disagreements. But why do they have to get personal and tell me you're this and you're that? You're an industry shill. You don't know. Why does it have to degrade? Well, I know why, because they have nothing to stand on, right? So I stopped engaging this one person. I'm just deleting, because if you say I disagree with you for this reason, okay, I'll leave the comment on, even though you may not be right. But if you start to call me names, that's it. There's no point in that. Because when you start calling me names, you've run out of arguments, and that's the end of it. Whether it's SARS-CoV-2 origins or the safety of vaccines, it's just a crazy thing where we've gotten to with science. Yeah, I feel very badly that we have to had to get here. But let me tell you something. I'm not giving up. I will continue to push good science with me and my team always. Oh, and the other thing, Kyrgyz Act. Oh my gosh. This is a YouTube channel, millions of viewers. They're very nice animations of science. They come out and say that we can't. It's equally possible that SARS-CoV-2 came from a lab. No, there's no data that it came from a lab. Kyrgyz Act. Maybe you can't figure that out, but it just goes to show that the nuances are lost on people that are not well steeped in science. They say the next pandemic will come from a lab. Really, what is the basis for that? Tell me of any epidemic that's come from a lab. We are very safe. We know how to do things carefully. So I'm very disappointed. I have to do a takedown of that episode because this is the problem. Millions of people watch it. A few 100,000 or so watch TWIV, and they win. Crazy stuff. All right, let's see. So we have Beyond the Noise. We have Kyrgyz Act. We have the TWIV episode. And then before we get to your chat, folks, I just wanted to remind you that the Amy Papers is still a thing. I've been updating it. And here are a couple of papers I wanted to highlight. First of all, here, yeah, it's looking good. Memory B cells. This is a review article in Nature Reviews Immunology. It just came out on the 3rd of July. It looks like there's no open access here, but we've learned a lot about memory B cells during the pandemic, and this is a nice summary of it. So if you have open access, check it out. Anyway, and the next one here, this one, I noticed that Kip just put up a link, rare link between coronavirus vaccines and long COVID-like illness starts to gain acceptance. So these are a series of studies where they think there's some evidence that the COVID vaccines are associated with some form of long COVID, which they are now calling long vax. Here it is, long vax. And, it's very, it's very rare, but it's starting to get some attention. I just want to bring out this statement from Peter Marks, who's director of CBER, which is where Amy Rosenfeld works. We can't rule out rare cases. If a provider has somebody in front of them, they want to take seriously the concept of the side effect, but he worries about the sensational headline that could mislead the public, and he emphasizes that vaccine benefits far outweigh any risk. This is the key to understanding on any side effect. That's very rare, and there are no numbers here. It's far rarer with vaccination than with actual COVID, so it still is a benefit to get vaccinated. But this is worth reading. There are a few case studies. Here's a single man who developed this POTS, as it's called, which is postural orthostatic tachycardia syndrome, which is something that you also see with ME/CFS. And I don't know how they actually know that these individuals didn't have an infection, an asymptomatic infection, unless they checked antibodies to, say, nucleic capsid protein. But anyway, this is something to look out for, see what's going on here. All right, so that's one. And then here's an interesting one. Genomic screening of 16 UK native bat species uncovers coronaviruses with zoonotic potential. This one looks like I could download right here, so it's open access. You can check this one out. So limited characterization of bat-borne coronaviruses in Europe. Actually, I would say limited anywhere, but it's certainly also in Europe. They screened 48 fecal samples from 16 of the 17 bat species breeding in the UK. They recovered 9 complete genomes, including 2 novel coronaviruses. And at least one of these can bind to ACE2 for infecting human cells, albeit suboptimally. So that's why they say it has zoonotic potential, because it can bind ACE2. But, of course, it ignores the fact that beyond ACE2, there are a lot of other things that can happen for a virus to have zoonotic potential. That's the first step. Obviously, if you can't bind a receptor, everything's off. But one of them has a 4 amino acid motif in the spike, which is 1 amino acid away from a furin cleavage site. They changed it to a furin cleavage site, but it didn't help spike cleavage. So just more evidence that we need to look and see what's out there. I think that's quite interesting. All right, here's one that Daniel Griffin discussed on the clinical report last week. Intrinsic and effective severity of coronavirus disease, 2019 cases infected with the ancestral strain and Omicron BA2 variant. This is in Hong Kong. Let me just go to the conclusion, right? Let's go to the videotape. Omicron has comparable intrinsic severity to the ancestral Wuhan, although the effective severity is substantially lower in Omicron cases due to vaccination. Let's just pause. Omicron has the same virulence as the ancestral strain. How many times have Amy and I said this? As study after study comes out saying Omicron is attenuated. And we said, no, you can't conclude that because of the way you're doing your studies. And now here they find that it is comparable. And the only thing that makes it milder is immunity. Daniel Griffin's been saying the same thing as well. I always told you, just wait until we get the right studies done, where the populations are as similar as they can be. And then we'll see what happens. Same thing with the damned transmission. That damned T word that I tried to get even Trevor Bedford to address, but he wouldn't agree to it. But he did say at the end that virologists and epidemiologists look at transmission differently. It's what I've been saying. It's what we've been saying all the time. Okay. All right, let's go back. I had a few more of these I wanted to tell you about. Here. Incidence is the last one. Incidence of myopericarditis after mRNA vaccination. This is a meta-analysis focusing on people 12 to 17 years of age. So they searched electronic databases and looked for people who had been vaccinated and developed myocarditis. Okay? So the incidences of myopericarditis after mRNA vaccination among adolescents were 43 cases per million vaccine doses for both BioNTech and mRNA 1273. 41.8 cases for BioNTech alone. More common among males than females, and more common among those receiving the second than the first dose. And here, this is the bottom. Here's the one I want to say. Oh, where is it? Where is it? Oh, all of them were significantly lower than myopericarditis in adolescents after infection, right? And it was another one. Where is that? There was another statement about other viruses, but maybe that's another study that I'm looking at. Anyway, it is quite low and far lower than in COVID. And remember, this is a treatable condition, okay? I think it was the long vax that there is some long vax in other vaccines as well. That's somewhere in here. Somewhere in there. Sorry, I can't find it. So it may not be specific to COVID vaccines, although they propose a mechanism that might suggest that it is. All right, let's get on to your questions, comments. Nicola sent an email to Dover Books. They reprint scientific books. Suggested to them to reprint John Paul's book on polio. That's a fabulous book, A History of Poliomyelitis. I hope they do. I'm not sure they will. There's not going to be much of a market. There is a used book market, but they're very expensive, of course. But I highly recommend it. It's a very good book. Nicola says it's 24C and dark in Italy. Yeah, it's 2 a.m., right? Yes, shameless plug for Twib mugs. By the way, if you go to cafepress.com/Twib, they now have mugs with this different colors inside that you can get. They don't have a small one in this shape. I really like this shape, but they do have a large one in this shape. But all of them have the coloring available inside, which is something I hadn't seen before. Renzo, hello. Good to see you. Renzo and I chat on Instagram all the time. He's somewhere up in the Toronto area. And he has Lise from Columbus, where we're going to have ASV next summer. That should be a lot of fun. Of course, Andrew is down there in New Zealand. Welcome. Khan, interesting. Thank you for continuing this, especially as Florida has so much anti-science. Yeah, it's unfortunate. It has anti-science because it gets votes, right? Not because it's right. They banned gain-of-function experiments. That's the other thing about the Kyrgyz Act. No gain-of-function has never, ever led to any issue. And they're concluding that the next pandemic will come from it. It's just people don't understand what a gain of function is. And we can explain it hundreds of times, and they still don't get it. So I will keep explaining it hundreds of times until I run out of breath, folks. You're not going to stop me. Eastern Long Island. Eastern Long Island. I'm going to go to Plum Island. They invited me to visit and do a tour on the actual Plum Island. That should be fun because they're closing in about a year. It's all going to be transferred to Manhattan, Kansas, as you may know. Yes, blue glasses plus spike. I really like this spike shirt. And it's a little chilly down here, so I have a jacket on as well. I know you can't see it because of the microphone. You got to figure out what to do. I need to have this mic here, and I don't want to put it over here. I guess I could do that, right? Does that work? Yeah, but you hardly see any of it. I need to raise the chair up. I kind of like the mic down here. To hell with the spike. Thank you very much. Glad you like it. University of Toledo. Hello. It's in Ohio, right? Welcome. MK is in Eastern Massachusetts, I think. Okay, here we go. Principal managers. So, the difference between virologists and epidemiologists when saying a strain is more transmissible, the virologist will need to see the lab work and the epidemiologist will go on genome surveillance. Okay, it's not as simple as that. The epidemiologist looks at the movement of a virus in a human population and counts cases and rates and so forth. And then they look at the genome and they say, the genome is different, so it must be more transmissible. So what if the genome were the same? What would you then say? Well, you would say that somehow people's behavior are influencing the transmissibility. And I asked Trevor that. I said, the genome does not tell you about human behavior. And no one takes that into account. He made some comments which I don't think really addressed it. But that's what a virologist would do, is do some transmission experiments perhaps that would say, no, it's not more transmissible. So the drive of the virus in the population must have been done due to human behavior mainly. Now, this is not perfect. I totally agree it's not perfect because animal models are not perfect, right? However, you need to do some experiments before concluding it's more transmissible. To a virologist, more transmissible means it has some property that allows it to transmit better from one person to another. That can be a property of the virus or it can be a property of human behavior or both, right? So you have to take both into account. And the epidemiologists don't. They say, this virus is spreading. I'll never forget alpha. We talked about alpha spreading in the end of 2020 in the UK, and they had just opened up from lockdown. So how do you disconnect the sequences that you think are driving transmission from human behavior? And they never tried to do that. They didn't even mention it. That's what bugged me among everything. They just concluded it's more transmissible. With each variant, it's more and more transmissible. I said this so many times. But virologists like Ron Fouchier, they will say we need to do the experiments to prove that. That's my take on it. What is constantly stimulating B cells to maintain a constant level of gamma globulins? So there are memory cells around, right, that are long after the infection or the vaccination. There are small number of memory. There are memory B cells, which can be in the bone marrow. They can be in the circulation. And then there are plasma cells, which are the ones that produce the antibody in high quantities. And those will just produce antibodies. They don't have to be stimulated to produce them. And they produce a low level, and therefore when you see low levels of neutralizing antibodies, for example, 1 to 8, you know that there's some memory there, as long as your assay is sensitive enough. Of course, it could be that your assay is not very sensitive, and therefore you can't really say anything about whether there are memory B cells or not. Good evening from Tomball, Texas. 30 seats, pretty warm there. Good to see you, Laurie. Okay, Dr. Bedford seemed to equate immunity with antibodies alone. Yes, most people do. So here's the thing. I just got a text from Rich Condit. Let's see. Okay. Talking about this Friday. If you have high levels of neutralizing antibody, that will impair infection, right? So that's what he's talking about when he says immunity. But if you don't have good neutralization, like the Omicron XBB, or not neutralized by antibodies induced by, say, older variants, you still have good protection against severe disease and death. Why is that? Because there are other forms of immunity besides neutralizing antibodies. But those are the ones that everyone thinks about in terms of preventing infection. And so they equate it with immunity. But immunity you have to define, right? If you want immunity against severe disease and death, it's also T cells and maybe complement NK cells, FC-mediated functions, and so forth. So not straightforward, not fair to really just say immunity is antibodies, yeah. Houston, Texas. I just thought there are probably a bunch of you in Houston. I was there Friday, Saturday, Sunday, left Monday. Should have hooked up with some of you. Too bad I didn't. Next time. We went to a bar. It was Mark Martin's birthday on Friday. And he said, I'm at a bar with a bunch of people. Come have a drink. It was a rum bar. I couldn't find it. It was in this part of town which seemed to be the party part of Houston. And I walked all over the place till I finally found it. It was very hard to find, but it was a good rum bar. And I said to the bartender, I want a Manhattan with rum. Can you do that? It's probably like a horrible thing to say, but she said, yeah, no problem. We could do that. And she made a good one. It was good. And Elizabeth says, a key thing to remember is that epidemiology can't separate the disease from the host population. Virologists study the virus independent of its host behavior. That's exactly right. Hello, Kip and Laura from San Francisco. Hi, an interesting piece in Science describing a rare condition, longvax. Like things like POTS are thought to be from rogue antibodies, which, like spike, bind to ACE2. Do you think this is happening? I really don't know. I mean, the model would be okay, but we have no evidence that there are such antibodies. So basically, if you make an antibody to spike, some of those could be against the receptor, and we used to call those anti-idiotypic antibodies. But I just don't know if we're confident that this is actually a thing in the population. I think we need to collect more data. I think it's possible. I don't know if it's happening. As I said, I don't know how you can separate. If you can ascertain that people aren't infected unless they're looking at antibodies. But I think more data are needed for sure. And that was the paper that I brought up earlier. So thanks for mentioning that. Question from the Koistia Symposium, Part 1. I know not a lot of people have watched at Koistia Symposium, but they're good talks. I mean, they're standard science talks, right? What's the difference between MOPV and trivalent? So M is monovalent and T is trivalent. So poliovirus exists as 3 types, serotypes you could call them. They're defined originally because if you had infected an animal with one type, let it recover, and then challenged it with another type, it wouldn't be protected against disease caused by the other type. So we used to vaccinate against all 3 types, although many countries still vaccinate against all 3 types. Like the U.S. inactivated polio vaccine is all 3 types. Anyway, monovalent is just one serotype. In this case, it's OPV2. And trivalent is all 3. So we used to use trivalent OPV up until 2016. This is an interesting story. And at that point, 2 things happened. First, we didn't see any more cases of polio caused by type 2 and wild type 2, and the WHO concluded it was eradicated. And there were still a lot of cases of polio caused by vaccine-derived type 2. So they said, let's take the type 2 component out because it keeps causing these outbreaks of polio. And they took it out, but unfortunately, they had neglected to see circulation of type 2 poliovirus in the northeastern part of Nigeria, Boko Haram, is that how it's called? And that virus, they went in to try and stop that outbreak with, what do you think, monovalent OPV2, and they reintroduced it, and that virus is circulating to this day and causing hundreds and hundreds of cases of polio in Africa and a few other countries. So that's the difference. M is just one serotype, type 2. Trivalent is all 3. Hope that helps. Yes, scientist versus PowerPoint. That's pretty funny. I thought it would get more views than it did. And I guess the reason is most people don't use PowerPoint, right? So they don't know the things that can happen. So in this case, I had my laptop on the podium. We were running a Zoom session on it. And the slides were on the laptop and the Zoom session was on the board. So if you wanted to get to the next slide, the pointer had to be on the laptop screen. And if you didn't, it would go, it would make that noise, and that's what scientists versus PowerPoint is all about. Oh, boy. Yeah, maybe it'll get more. I don't know. John, 496 and 500 were good episodes on vaccine hesitancy. They lined up nicely with Beyond the Noise 7. Yeah, that's a good point. Let me pull those up. TWIV 496. TWIV 496. So that's vaccines work whether or not you believe them. This was actually quite good. We let's share the screen for that one. Rich and I went to Irvine. And we had 3 vaccine people, Stacy Schultz, Cherry Douglas, Dikema, and Andrew Neumeyer, talk about vaccine facts and fiction. That was very good. And then the other one is 500. So let's look for TWIV 500, see what... Oh, I know what that was. That's the one we did in Austin, Texas, with Jenny, TWIV 500, who's a pro-vax person in Austin, Ginny Su, and she's she got her law degree at Columbia, moved to Texas, and is a pro-vaccine activist. And she talked all about vaccine hesitancy. Let's see if her web page still works. Immunize Texas. So this is the organization she co-founded. Non-medical vaccine exemptions are on the rise in Texas. That's really a problem. Some of these states doing things like this, right? Really a problem. I don't know what people are waiting for. What do they think? We're going to have outbreaks of infectious diseases that should be all preventable. Marco says the short videos you do often are wonderful, but the latest one I enjoyed because it explained the difference between the virologists obtain information versus epidemiologists. Yes, epidemiologists observe, as we said, populations, and virologists do experiments, right? They observe the results of the experiments, but they're actually intervening. But when you're doing observations in populations, you don't intervene. You just say, oh, there's this rate of polio in the population, and that's it. Yeah, that's a good point. So these people on Instagram, I can block them, yes. I think it was yesterday that person made 5 or 6 really nasty comments, and I just deleted them all. but I need to know the name before I could block them. And I'm not going back to the older ones because, I find it painful to read nasty comments. If you look at Beyond the Noise 7, over 1,000 comments. Oh, my gosh. And many of them are just nasty. I can't handle that. Enough nasty in the world. So if that person shows up on Instagram, I can block them. Yeah, I can do that. I don't want to do that for most people, right? Up until a few days ago, I think it's a she, but I'm not sure. She was just disagreeing and said I needed to get experts on TWIV. Robert Malone, a virology expert. No. I felt like saying Robert Malone would not pass my virology course, but I didn't want to say that because that gets more inflamed, right? Although it's funny because he probably wouldn't. No, he's not a virology expert. He's got his own opinion, which is not right, but he's not a virology expert. So that person said it with respect to the UPenn episode, from Nature, not a lab. Said you need to get the other side. No, I don't need to get the other side. I have no facts. TWIV is fact-based. If you have facts about lab origins, I'll get you on, but you don't. I get people on who say, here's what we have for zoonotic origin. And I have no shame about that. And I tell people, look, I've been studying viruses for 40 frickin' years. I don't know everything, but this is a zoonotic event. It was a zoonotic event. No, they have names on Instagram. I don't know if they're real names or not, but it doesn't really matter. I mean, I could block them if I see them next time. RFK Jr. was paid by the Kremlin to produce videos for Russia Today's RT America network for over 10 years under the show name Ring of Fire. The Russian trolls have gone wild on that video. So, yeah, Pull Off it said that... So Andrew Wakefield was taken down by investigative journalism. And so that's what has to happen with RFK Jr. if we think he's a threat. So, I mean, as Paul said, it may not be a viable platform to run for president and be against all vaccines, right? Because that's just it's an untenable position. You can't say things like the 1918 flu was started by the flu vaccine. That's just absurd. And it just shows you how little Rogan knows that he didn't say, wait a minute, RFK Jr., there were no flu vaccines in 1918. In fact, we had not even isolated influenza virus, so we couldn't make a vaccine. No, but it gets headlines, and that's all that matters is getting headlines and getting a lot of views and making money. I mean, I bitch about not getting a lot of views, but we just keep going because that's the way it is. Don't mention the trolls. Yeah, well, I didn't mention their names. I'm just complaining. This is a safe place for me to complain about them, right? And Steph gives me instructions for blocking. Yeah, I can block them. No problem. Don't feed the trolls. They try to provoke me. Yeah, they do. I'm reacting here to you folks because this is a safe place, but I typically don't respond to them. Yeah. And the last twin, it was mentioned that translation could take place in the axons of hypothalamic neurons. Without axons having cytoplasmic rough ER, how could this be possible? Well, that's a good question. We never knew that translation could happen, but it's been shown that there is protein synthesis occurring in axons. And so there must be ribosomes there because it's not happening without ribosomes. And a number of people have made that observation in various kinds of neurons, including Lynn Enquist, who used to study neurons in culture infected with herpes viruses. Said, yeah, there's translation in the axon, contrary to what people used to think. Yep. Yeah, Kyrgyz Act is disappointing. That is Philip Detmer, who wrote Immune, and I'm very disappointed in him. Now, I'm not going to say I'm not going to listen to you anymore because of one podcast, right? That's what people say to me. I don't like your this episode. I'm not going to listen to you anymore. I'm gone. I'm not going to say that. You should know better than that. You should know better. You're just listening to the loudest people to get your facts. You're not investigating. You're not reading the papers where there's evidence for a zoonotic origin. It's too bad. Yeah, this is right. It's hard to win an argument with someone not constrained by facts. That's why no one should go and debate these individuals, right? No one should go debate them. I just dread the day that someone, a reasonably influential scientist, decides they're going to debate them because, I don't know, they want attention or $100,000, and it will be an S show, to paraphrase someone who is often here. Climate scientists get called shills and all sorts of abuse too. Yeah, I get called a pharma shill. I don't get money from pharma. I purposely did not take money from Pfizer to do podcasts because of this. I said, no, I don't want money. I will say the truth. It's fine. And people will say, well, you're still a shill. Okay, make up your own terms. It's fine. No problem. Glad you liked it. Carol, our favorite nephrologist. Thank you. I'm glad you liked it. I knew it would be good. I posted it right away. I edited it that same day and put it up there. Had 1,000 views in an hour. That you can tell it's going well. Oh, there a website that tracks wastewater surveillance. There is one. There are several. I can't think of it right now. I heard this in multiple talks at ASV. Someone's going to know. Someone's going to know. I've been having fun reporting the anti-vax YouTube commentators who harass and bully the video hosts. They're just ridiculous. But we will keep going, right? We're not going to stop. I recently viewed the Bill Nye Ken Ham debate on the Creation Museum. Again, I feel it's hopeless. Dark Ages is upon us. Although, Offit said at least it was civil, right? They both felt secure in their positions and stated it. But for it to deteriorate is not good, right? Kent the Cat Guy, thank you very much for your contribution to science communication. So I don't mean to beg folks, but the best way to combat this is to support channels like MicroTV, where we give you the facts. I mean, people support Kyrgyz Act, and that's great because most of the time they're good. But when it comes to viruses, they shouldn't be saying anything. I mean, they can go look up stuff on the internet and think they're learning, but they're not virologists. Check me if I'm wrong. If there's a virologist on the panel, then, boy, you got your facts wrong. Or you decided to purposely make them wrong to get more views. Dr. Bedford's story about getting to viruses was very interesting. And he was a particularly articulate guest. I watched a few times. Thank you. I'm glad you liked it, Marco. I always have fun picking guests at meetings. You could pick from a lot of people. I thought he would be good. And in general, I thought he was articulate. Yes. He was a little nervous, though. It was funny. we were trying to do a sound check ahead of time. And I said, talk. And he said, well, I don't really have anything to say. I said, okay. What did I ask him? Why did you decide to come on TWIV? And he said, well, I thought it was the right thing to do. I said, okay, anything else? And nope, that was it. It was very funny that he didn't want to talk anymore. But I thought, yes, I agree with you that he was a good guest and presented most of the science really clearly, and I liked it very much. Yeah. Okie dokie. I wanted to show you a picture here. Let me get it first. Sorry, I'm delaying here a bit. I want to go back to Quebec City. Where is Quebec City? Hold on. Nope, it's not there. I'm not finding it. Okay, let's move on to the next question. Luis also liked the approach at frequency and evolution, change on frequency as an idea of fitness. Yep, fitness. And notice how many times he said fitness. Do you guys remember who kept saying fitness at the beginning of the pandemic? Let's not refer to transmissibility yet. Let's talk about fitness. Yep, it was us. Claire says, for polio IPV, which brand product is a good option in the U.S.? Well, there's only one available, I believe. Let's look. IPV. I was just looking at the product, the FDA package insert. Let's see if we can, no, IPV vaccine. IPV is also innate partner violence. So it's the only one available in the U.S. Who makes IPV vaccine? I'm not going to say Sanofi Pasteur. That's my guess. But let's see what the great Google. Sanofi. Yes. So Sanofi Pasteur is your only choice in the U.S. for IPV. But your question is, you got it 26 years ago. Should you get a booster? No, you don't need to get a booster. My last IPV was in, I got IPV in 1955, and then I got OPV in 1962. So 62 is over 26 years ago, and I'm not getting another booster. It's not needed. I think the immunity is quite durable, so I don't think you need to do that. And I'm not giving you medical advice. A lot of people will come on and say, Vincent, what are you giving medical advice for? I'm just saying that potency of the vaccine is sufficient so that a booster isn't required. I'm not going to tell you what to do, but you can listen to what I say. How's that? Okay. Oh, boy. If they had wastewater surveillance in Wuhan and could pinpoint the first cluster and administered antivirals, then no pandemic. A lot of people will disagree with that, but I have always said that if we had a large stockpile of antivirals and we started using them early, you could blunt transmission and perhaps blunt the pandemic. Yeah, I mean, you would certainly avoid a lot of deaths, right? And by the way, Bedford is predicting, 80,000 deaths a year in the U.S. in 2023, COVID deaths. And I asked them, they're all preventable with antivirals. She said, absolutely. So if you could get to those people and give them antivirals, it would be all preventable. But we're not. We're not doing it enough. But so I think at the beginning of the pandemic would have made a big impact for sure. Yep. But it's hindsight, right? You'd need enough. You'd need enough antivirals to be able to treat everyone. And it's not clear that we would have had it, even if we had a pan-coronavirus antiviral. How many doses would we have stockpiled? I think we stockpile 100 million smallpox doses in the U.S., antivirals. Could be wrong on that. So just for the U.S., you'd need a lot more to stop a pandemic globally. So I don't know if we'd have enough. That's the problem. They have a shelf life, so you can't keep them forever and so forth. And who's going to pay for them? That's the problem. Craig says, is there any way to show that the exact people who developed myocarditis from the vaccine would have developed it from infection? The exact same people? I don't think you can do that. You have people who have gotten infected before being vaccinated, right? And they have developed myocarditis. But if you're worried that it's a genetic predisposition, right? So that if the vaccinated people had also been infected, would you also see it? Would you see it predominantly in them? That's a good question. I don't know what now we couldn't do that kind of study, right? Because if you get, unless you have a population of people who got COVID, and then you see if they got myocarditis, or vice versa. You could see if they got myocarditis both times, right? But I've never seen that sort of study at all. So I think it would be really hard. Someone on the Discord suggested Biochemistry Explained, a practical guide to learning biochemistry by Thomas Millar. I went ahead and bought it when I found it used for $9.99. Okay, I haven't heard that one. Let's look. Biochemistry Explained, Thomas, what was the name? Millar. A practical guide to learning. Well, I'm not familiar with it. And you got a good deal because here at the publisher it's $50. So, good deal. Let us know, okay? I think that's a good thing for you to do, if you're so inclined, is to read it and see what you think and report back to us so that other people who are looking for a biochemistry book can go for it, right? But you have to recommend it. So, do you think it's necessary after having a bivalent in October to have anything else? So just you had a bivalent, which is ancestral plus some early middle vintage Omicron, right? Do you need anything else? Well, it depends. I mean, are you at high risk for severe disease? That's the question you have to answer. So Daniel and Offit will both say, if you're at high risk, over 75. And boy, last time someone yelled at me for saying, why are you moving the bar up? Because you're 70? Do you want to make it 75? Oh my gosh, no. So leave me alone. 75 and up, comorbidities, other healthcare problems. In that case, any infection is going to put you in the hospital, or it may put you in the hospital. So it would be good to get a booster. And you can get that in the fall, right? That's the idea, is that the FDA recommended, the FDA committee, advisory committee recommended, BA 1.5 or similar, 1.1.5 or similar single composition booster in the fall. And you might want to do that. I won't be getting it because, not because I'm less than 75, but I don't have any comorbidities. I have been infected last November, which would be an early Omicron variant. And I really think that's sufficient. But more importantly, if I test positive, I will take Paxlovid, because last time it worked really well. So that's what I think. But I'm not a doctor. I'm not a medical doctor, so don't get medical advice from me. But virologically speaking, I'm just repeating what Paul Offit and Daniel Griffin say, because I can't give medical advice. But virologically, the neutralizing antibodies induced by October's variants are not going to be great at neutralizing the latest Omicrons. And so in a certain population, that could be a problem, but not in everyone, right? The mic blends in with the T-shirt. Yeah, but you can't see the spike, right? It has to be lowered. It should be tilted down a bit, but I can't do that. My camera is not easy to tilt, but and actually, let me fool around for 1 minute. Hang on, camera controls. Where are the bloody camera? Here we go. Camera effects. I think I can do something here. Nope, I can't do that. I can do zoom and pan. No, but that's not helping. All it does is make it smaller. So we'll turn that off. Is there a tilt? Nope, there's no tilt. I would have to move the camera down. So I think it's worth it, right? If we're something like this, but then my head would be cut off. I'll work on it for next time. Yeah, T-shirt. All right. There was a TWIV on phages for resistant bacteria. Have there been any advances in treatment for antimicrobial resistant bacteria? So, yeah, we did a TWIV 500 in Austin, which was it 500? 501, which is in Texas A&M, which was all about that. But I just got an email today from someone who said there's another case of someone whose bacterial infection, antimicrobial resistant, was taken care of by a phage therapy. Let's see if we can pick up the news story here. Here we go. 1 day ago. Oh, this is a Canadian trial. Right. Let's share the screen. Okie dokie. Here we go. First Canadian trial successfully uses phage therapy to stop a life-threatening urinary tract infection caused by a superbug. You are considered clinically and biologically cured. 72-year-old retired librarian. Had multiple urinary tract infections. It was treatment-resistant E. coli, also known as a superbug. Okay, so what did they do? She became patient number 1 in the first Canadian study using phages. Unity Health in Toronto. Oh, look at this. Phages shoot to kill. They have a natural ability to go to the source, make more of its source, and continue to be there while the infection is there. Okay, fine, Dr. German. And so they gave her phage targeted for her bacteria. And there she is, I guess. The doc and the pharmacy manager. And she was cured. Stephanie Strathdee. You guys know who that is, right? She's the wife of the guy who was cured by phage treatment. He had a Acinetobacter infection. And so she investigated and found phage therapy and pulled all the strings to get it done. Now she has a book about it, and she goes on speaking tours. And she spoke at ASV, actually. Paid her a lot of money to speak. That's what authors do, right? I go speak and I get $200, which is fine. An author speaks, and the author say, "Well, Vincent, this is what we do for a living. You do other things for a living." I'm not going to begrudge you your pay. That's fine. How do we have for the likes here? We have 195 people, 131 likes. That's not bad, but you could tap the like if you're near it. Please do it. Louise, thank you so much for your contribution to science communication. Setting the record straight, right? That's a good way to put it. That's what we like to do here. And I'm glad you all come, all 200 of you. It's a nice little community, and I appreciate that you want to hear what I have to say. Claire says, for COVID mRNA vaccine, growing evidence for the vaccine to prevent long COVID with shorter interval between vaccination and natural infection, the better, versus interval should be longer for best protection. I'm not sure that the interval is really set in stone yet by these observations, but I agree that vaccination does appear to reduce the incidence of long COVID. But about these intervals, yeah, there are some suggestions, but remember they're observational studies and they could be confounded, and so I would wait for them to get more data. I understand him saying that the ratio of strain prevalences show the relative transmissibility. The ratio rather than absolute adjusts for behavior. Yeah, but that I don't think that's enough to do that, is it? My opinion that how can you the ratio of what? The ratio of strain prevalences show the relative transmissibility, or the ratio rather than absolute adjusts for the behavior. I'm not sure that that's correct. It's assuming random exposure, I guess, right? Was Houston hot and humid enough? Yeah, it was fine. I have no problem with hot and humid. I like it, being a Mediterranean origin, right? So it was fine. I enjoyed it a lot. I didn't mind going outside. It's good. Yeah, even Athens was hot at some parts of the day. So yeah, but ASV will be in Columbus in 2024, and I will do my best to get everyone in who would like to go in, and just at least for sure, she has some friends coming, and it would be fun to do a meetup afterwards of all the TWIV fans, outside of the meeting. That would be fun. I'd happy to go out with you guys. Plum Gut. Yeah, I know about Plum Gut. I've heard of it, right? I've traversed it. The water between Orient Point and Plum Island. I had a rough small ferry ride through the gut one year. So when I was a graduate student in the 70s, I made a few trips to Plum Island to do some research. And yeah, here's Plum Gut, and it can be rough. It was okay for me. But I am going to visit one last time to do a TWIV. I met someone from there who's probably not listening, but I have to get in touch with you. Who's the director? Director of Plum Island. Let's see. Larry Barrett. I don't think that was the guy who said hello to me. So whoever it was, how am I going to get in touch with them? I know some people on the island. I can email them. Yeah, I'd love to visit and do a final TWIV out there. Yeah. Myers is the best rum in my opinion. Well, she didn't use Myers rum for her Manhattan. She used something that I'd never heard of. I mean, they had hundreds of rums, right? She had to climb up on a ladder to get this one rum. 7 Saddles or something like that. Is that a rum? Let's look it up. 7 Saddles rum. No, it's not a rum. I forgot. Should have taken a picture of it. No, anyway, it was something else, but it was very good. I'm sure Myers would be okay. But she said, let me get the right rum for a minute. Rumba is a dance. I think so, yeah. I'm not the one to ask. I'm not a dancer. Yeah, it's ballroom rumba. You bet. What would you tell a teen who doesn't want to wash hands? The teen says it builds their microbiome. Yes, you can wash your hands to prevent transmission of viral respiratory infections, right? But you will still have a microbiome because you're not going to wash enough to get rid of it. So I would aim at something in between. Something in between to reach a compromise, right? Because it's clear that certain respiratory infections, like rhinoviruses, are transmitted by contaminated hands, right? So that will prevent a lot of that. So you say, okay, we can prevent that, but you don't have to wash every hour, so we'll keep your microbiome. But it's true, but remember, the hand's not the only source of the microbiome. We have it in other parts of our body as well. Vistotuti, could gene drive wipe out malaria mozzies, malaria mosquitoes? Is there a TWIV on how gene drive works? We have done a couple of TWIVs where we talk about using Wolbachia to eliminate dengue, right? So releasing Wolbachia-containing mosquitoes, which is kind of a gene drive because you're driving Wolbachia through the population. But there are other forms of gene drive also, which I heard actually a couple of years ago at ASV. I heard a guy give a talk on it. It was really good, and I always wanted to have him on TWIV. So I think that's a good idea. Let me write it down. Unfortunately, I don't have my my trusty field notes. So gene drive in mosquitoes, TWIV. Okay, we'll do that. Written down on the piece of paper. It's a good question. Rumba. Hello, Vanity Nutrition. Welcome. I don't know if you're still here, but welcome, and thanks for popping in. I like the look on Amy's face in one clip. Yeah, it was pretty good. I have some videos of the dance at ASV that we're going to show. So my new assistant is Karen. She's come with me on a few trips, and she runs the equipment. She comes to the incubator a lot, and she took video of the dance floor after the ASV banquet. It was a lot of fun. So we'll release some of those for you to see. Scientists being normal people, right? Dancing and having a good time. I tell you, the young ones, they're really into it. Cool. The stuff they do is very cool. What are the differences in the genome structure between the three types of polio? Oh, you want to know the percent homology difference. I don't remember offhand. Percent genome difference, polio serotypes. If Amy is listening, she knows. Why can't Google give me a straight answer? Of course, it depends on the part of the region that you look at. No, that's not a good one. Of course, the other issue is that you have to have a pure type 1, 2, or 3 without recombination, right? I don't know the answer of the homology. Do I dare to? Oh, let me ask Amy here. What percent nucleotide difference defines polio serotype? Let's see if she will answer that. Okie dokie. Thank you, Nicole. I will get back to you. Yeah, here you go. There's a gene drive on dengue. Yep. Another takeaway from the last TWIV, SARS-CoV-2 is 3 times deadlier than flu. Average 80,000 excess deaths per year. There could be less with vaccination and antivirals. That's correct. The 80,000, and I asked him, and he said it's totally preventable with antivirals. Yeah. Yep, you got it right. So Bedford gave a talk earlier in the conference, and I took a lot of notes, and I used those as the basis for my questions, right? Because I wanted you guys to hear what he'd talked about. So I asked him to repeat a lot of the stuff that he had said in his talk, and one of them was this 80,000 excess deaths. Yeah. John says, it's amazing that people will attack pediatricians and researchers dedicating their lives to improving health and not question a lawyer trying to... Yeah, it's sad, isn't it? Because, yeah, for the most part, scientists and physicians and public health people are trying to save lives, right? They're really dedicated to that, and then you attack them, and it's not really fair. You can disagree with them, as I said earlier, but you shouldn't attack them. A lot of trolls have moved on to the AI will kill us or save the world debate. Anyone who knows anything is a target. Okay. Any kind of progress is not good, huh? Well, that's out of my lane, of course. But I see a lot of good being done, but I can see a lot of bad also. A lot of things are out of hand, yeah. Tom says, besides facts, you need solid, plausible argument, and they don't have that either. No, that's correct. Everything is inconsistent, internally inconsistent, right? But they don't see it that way, and they can just spew nonsense, and people will buy it because most people are not very critical, right? Which is too bad. It's not hard to be critical. It just takes a little thought, but people don't want to be for whatever reason. How do we combat RFK Jr.? He's getting a lot of coverage. Yes, he's taking advantage of his name, right? He wants to be a presidential candidate. I don't know. As Paul Offit said, he's probably a good target for investigative journalism to find out who's paying him and why, because he's getting paid, and that could do it. But I plan to just counter everything he says, and Dan Wilson will do the same. I think it's what we can do, and every scientist can do, because you're right. You're going to get a lot of people thinking that he's right. But why would a lawyer be right about viruses, right? If you want to learn about viruses, who would you ask? Would you ask a lawyer? Now, I don't mean to impugn lawyers. You guys and gals are great at what you do, or can be, but you don't know about viruses unless you're specifically working on it. I knew a lawyer years ago who I worked with on polio litigation, and he knew pretty good things about viruses. Know everything, so that's why he hired me to advise him. I didn't take any money, by the way. Worked pro bono. And so there are some who know a bit, but it's a restricted knowledge and not a broad knowledge, certainly not of all vaccinology. But if I have questions about vaccines, I ask Paul Offit, right? And to impugn him, to say he got paid $186 million from Merck, this is ridiculous. It's just an effort to besmirch his name, and it's not even true. But Rogan's not going to challenge him. YZ, Moderna full spike. No, Pfizer is spike also. No, Pfizer is full spike. They did try an RBD only in the beginning, but it didn't perform as well, so they went to full spike. So, no, they're both equally good at inducing T-cell epitopes. They're both full spike, yeah. Yep. Why do you think RFK came out of the gate so hard on Paul? Immediately called him a liar. I was shocked. Because he has nothing else to do. He cannot... What is he going to say? Yeah, you're right, Paul. Because Paul was right on every point. So he says you're a liar. That's what they do when they have no facts and no internal arguments behind them. They call you a liar. When you think about it, what are you going to do? I'm not a liar, and this is why. It just goes back and forth. It's a ridiculous argument. But that's what I think. He just has nothing, and he knows it. So a good fraction of people will believe you if you call someone a liar. We've seen that in politics forever, right? So that's why he does it, and that's why he's going to use it forever. Yeah, you're a liar. So that's the same thing. Jeffrey Sachs, when confronted with the data on SARS-CoV-2 origin in the Huanan seafood market, he said, you're lying. That paper is a lie. This is not my kind of science to call someone a liar. You can disagree and say, oh, for this reason, here's a flaw in your calculation. But no, because he doesn't know. He doesn't know how to look at the paper. He can't even read it and understand it. So he calls you a liar. So Sachs did the same damn thing. I would say to RK, it's not possible that 1918 influenza pandemic was caused by a vaccine because we had no flu vaccines and we had no influenza viruses to make vaccines with. And he would say, well, you're a liar. We did have flu vaccines, and there are good records of it, and you're just lying. And so people hear him say there are good records of it or something like that, and they'll believe him. Racaniello's lying to protect the industry. That's what we're up against. It's a real pain. There's a quote I like. It takes years of work to become an overnight success. TWIV is a rare gem. Ian, it's very kind of you to say that. By the way, we are using your Bluetti. Yeah, it's a Bluetti in the incubator. My assistant Karen is all excited about it. She set it up and we're using it. It's very kind of you. I appreciate it. We've been doing TWIV since 2008, and I've always been very patient. I said, we'll get our audience slowly but surely, and we're working on it, and we're going to continue to work on it. And I do think we're a rare gem because we are a science program where we have scientists on the program, and they speak facts. They don't lie. They have no agenda. And they're nice, except when I'm not nice to Dixon, right? But that doesn't happen anymore. I know that was not good. But I think we are rare, and not enough people acknowledge us. So news outlets don't acknowledge us for the most part. Why not? Why don't you say, okay, here's our story, and if you want more information, check out this amazing podcast. No, they have to protect their own wealth. They have to protect their income, so they can't reference anyone else. So all of the papers, all the journals should be referencing us appropriately, but they don't. Now, I'm not bitter about that. I just don't understand it, because if you have a in-depth program that addresses what you just did in 2 minutes, why not, for people who are interested? And here, Dan says, that's true, but not in the forums that are Hotez was a bit of an exception. He responded strongly. I'm worried by RFK Jr. in the New Hampshire primary. There's a vacuum. Yeah, it's a problem. I mean, if this guy got elected, man, that would be disaster. for kids. Ridiculous. I am an educator, plain and simple. That is what I am. I am not a politician. I'm not a lawyer, not a doctor. I'm an educator, and I love it. And I have found new ways to educate, including this live stream and podcasts and lectures. And I think there's more to be had. I'm full of energy, and I'll do it till I drop. Believe me, I will. 24 Mac, we talked about this long vax. I've read it, and it's part of the Amy Papers list today, and I would echo what someone in the article said. We have to pay attention to this and make sure it's real, because in some countries they don't see it, in other countries they do. So we have to have more studies that look at it. But, I mean, it's very, very rare, and much more rare than long COVID after infection. So that's the good news. In my genre of environmental commentating, we battle the Heartland Institute, who's backed by Koch money. There's a lot of misinformation all over the internet. Things are not so great. I'm sorry. Yeah, we're all sorry. Why people have to do misinformation. I just don't get it. That makes them wealthy or gives them income. Yeah. Patrols will come in and tell us that CO2 is good for plants, that the greening of the planet is a good thing, not understanding the dynamics of Arctic and Antarctic ice. Yeah, trolls. Yeah. Yeah. Coffee House, hang in there. Thank you, and Dr. V, for being light. So, well, thank you. Appreciate it. John, thank you for your contribution to science communication. On a positive note, old TWIV is accurate and predictive, a worthwhile journey. I really liked Kate from NASA, and she returned, so looking forward to that. Yeah, so Kate was on first with me and Rich Condit at ASM microbe. I don't remember where it was recorded. And then she came on from the International Space Station. That was pretty cool. And something that none of you will see, at the more recent ASM microbe in Houston, she interviewed Andy Weir in front of an audience, which was entertaining as well. Could you share more about why two lineage could not be explained by lab leak? Well, it would explain, we always like to have the simplest explanation for things. The simplest explanation is that they came in two or more different animals. And in fact, there's evidence that there were multiple spillovers in the market. And I'm going to have the guy who did those calculations on TWIV. Let me write that down, too. So many people I have to get on. So the easiest explanation is that multiple animals brought them in from the countryside. If it's from the lab, then someone would have to come from the lab, get contaminated in the lab, bring it to the market. And then someone else would have to come from the lab, get contaminated with another lineage, and bring it into the market. And this is just unlikely to have happened because there are far more opportunities for spillover in animal, many, many more animals out there, non-human animals, than people in laboratories where, I don't know, there were 10, 20 people working in that laboratory. That's it. That's all you have. And the numbers just don't add up. And Eddie Holmes said it very nicely in his TWIV a couple of episodes ago. I should make TikToks of some of these. Oh my gosh, there's so many things I need to do. There's so many TikTok moments where he explains why, why one, the two lineages are just not consistent with a lab leak. I got to clip that out. Yeah, let me write it down here. Holmes. Now you could do many TikToks from that. I'm not really good at posting them. I can't figure out how to put a nice cover on them. TikToks. Yeah. Thanks, Claire. Could you have Naomi Oreskes, who wrote Why Trust Science, as a guest? Naomi Oreskes. Let's see. American Historian of Science. She became professor of history, blah, blah, blah. I think it could be interesting, right, in terms of why we should trust science, right? I presume that she has a good argument for why we should. I don't want someone who's going to say don't trust science because that's ridiculous. Look what science has done for us, right? So I'll write the name down. Okay, you got it. Pete, we've been so used to the studio sound quality of Zoom TWIVs, it's harder to hear on the live lecture hall discussion. Somehow it doesn't quite capture the atmospheres. What are you complaining about? You don't like the sound quality? I'm sorry. The rooms are never perfect, right? I think the Switzerland one was very good. That room was made for concerts. It was beautiful acoustics. The last one in Athens was a crappy conference hall with the bloody speakers in the ceiling, those things that are made just to play crappy music. And they pumped our voice through that. It sucked. It absolutely sucked. So that's why the sound is no good. We're at the mercy of the people who get us the room, right? We can't say we want an auditorium with a nice sound system. No, you're just TWIV. We're not going to give you that. So until such day that we can command better venues, we're going to be stuck with that. So I'm sorry about that, but just, try and slog through it because there's good content. Yeah. Thank you, Doreen, for your contribution to science communication, and love this Wednesday evening virus clatch. It's great. Studies now confirm that natural infection and COVID vaccines produce similar levels of protection. I have to say I'm tired of being called anti-vax just because people forget about basic science. Yeah, they're both. I mean, natural infection may be better because you've got more T-cell epitopes, right? But you don't want to make people say, oh, that means I don't need to get vaccinated. Although at this point in many countries, right, there's a high fraction of people that have either been vaccinated or infected, so it's a moot point anymore. But that's correct, Raphael. Infection gives you really good immunity. You should check out virologist Benjamin Neumann. He's been studying coronaviruses for 26 years. Okay, I will look up Benjamin. Of course, it could be Newman, but, the microphones are called Neumann microphones. Do you guys know what Neumann microphones are? These are like really expensive microphones. They're not the most expensive in the world, but here's an example of one at B&H. There you go. Look at that one. $4,000, $3,500. I would not get one of these, okay, because I don't need it. These are condenser microphones, which will pick up every sound in the area, and it will sound terrible. It's for if you have a really isolated booth where you're recording. That's great, but I don't have that. I have a $400 mic. It's great. It's good enough. But that's why I said Neumann, because that's how you say those. I will check him out, Garth. Definitely. There was a plan to have certain islands as safe places during a pandemic, for example, Iceland and the South Island of New Zealand, but with modern travel, this idea seems to be unworkable. Yeah, it is unworkable unless you want to not let people travel, which I don't know. I mean, your country was closed for a while, right? New Zealand. How did that work out? Can you ask Daniel if there are going to be more antiviral drugs being produced? Different kinds, yeah. I have heard multiple talks on different kinds of antiviral drugs. I sat at lunch at ASV across a guy who had a coffee mug that said the Molnupiravir team on it. So he worked on Molnupiravir at the Emory Institute for Drug Development. So if you remember, the original name of Molnupiravir was EIDD with a number. Emory Institute for Drug Development. So he works there. And he said they have more stuff coming out, and others do as well. So yeah, there are, I can tell you, I don't have to ask Daniel. There are more coming out. Yeah. I feel like we are in a Planet of the Apes movie. We are the chimps, CDC are the orangutans, and RFK Jr. Rogan types are the apes. That's a good one, Principal. I like that. Very good. Very good. And Ariel has the same T-shirt as this. Yeah. It's very cool. I just ordered a couple more today, so you'll be seeing them here on the streams in the coming week. So my assistant Karen said I need to match my glasses, so I got a blue shirt to go with it. I got some TWIV stuff. cafepress.com/TWIV. We have some good stuff there. Here, let me show you. TWIV. Let's do this here. It's a very nice. I've been cleaning it up lately because it got a little disorganized. So here we go. We have front page. You have fitted cut clothing. Okay, so this is the verbiage I was encouraged to use. Unisex clothing. Then we have caps, bags, mouse pads, drinkware, stickers and buttons, face masks. So all kinds of cool stuff here. Let's take a look at the fitted cut clothing just to give you a sense of what there is here. So we have, TWIV shirts of all kinds, short sleeve, long sleeve. We have hoodies. We have zips. Then we have the TWIV hosts, kind of a little thing that Vanity made with all the names, and then MicroTV down the front. Then we have, some of these are very interesting. This one is In Science We Trust. Mice lie, monkeys exaggerate. Life is an asymptote. We have Right Amy t-shirts. This one I particularly like. I just ordered this one, Countering a Miasma of Anti-Think. This was a letter from a listener a couple of years ago. I think that's exactly what we do. And they're all different styles. Here's Masks Are Cool, a little design by Sharon Eisern. We got Immune. We have Twin. I just added Twin today. This is a cool design. TWIP, Twim, and Tweevo. But what I want to show you is the mugs. They're pretty cool. Drinkware. Go here to drinkware. Now, let's pick a mug, okay? And then you can go white, black inside, white, navy inside, white, pink. You can do white, blue. You can do white, red. That's very cool. See, because my mug here, see, has a kind of purple inside to match the TWIV logo. But I think that's very cool. Don't you? Look at that. Let's see if it goes for the other ones. Let's see if it goes for the big one. No, only white for the big one. Anyway, I think this is cool. We make $1 on each item. That's all. So you can get your water bottle. You can get your mug, steel mug. You can get Masks Are Cool on it. A couple of other things. All right. So that's that's TWIV's MicroTV stuff, right? You got me on a little riff there, Ariel. Yes, phages were used in the old USSR, in Tbilisi, right, in Georgia. They started the first phage institute, and that exists to this day, for sure. Yeah. Mm-hmm. Thank you, Ben, for your contribution. S1 proteins are still showing up in my non-classical monocytes 15 months post-infection. Is it safe for me to donate blood? How do you know this, Ben? I'm just curious because you must have access to being able to look at that. I think it's fine. I think they're probably there from germinal centers that hold on to antigen for a long time. And I don't know what the levels are, but I'm not sure that it's a problem. Alan says, given a large enough population, a racial comparison should even out random differences in behavior, but the numbers would show fitness, which doesn't rule out intransmissibility. If it doesn't rule it out, I agree. I would call it fitness and then try and get some evidence for transmission. Now, the Omicrons, remember, are immune evasive. And so I would say that contributes to transmission because if a virus goes through the air and gets into your upper tract and the antibodies don't neutralize it, then it's going to transmit. So that's a transmissibility determinant, being immune evasive. But and we have those data, but we don't have similar data for all the variants. Yeah. Wolbachia bacteria released in Townsville, Australia. Yes, we did that paper on TWIV a long time ago. Glad to hear it's working out. When I was in Singapore, in 2019, I did a TWIV at the place where they make the mosquitoes containing Wolbachia, and they pack them up into little buckets and they send them out and they release them. It was a very cool discussion with the lady who runs the place. Yeah. It's never a good idea to drive any species to extinction. You never know how it will affect the ecological balance. Yeah. What about targeted local extinction like they're trying to do in this place in Australia for, mosquitoes that harbor dengue? I understand what you're saying, Andrew, but you have to balance, the pros and cons, right? And yeah, I would say if you want to get rid of all the mosquitoes globally, that could be a problem, but some local extinction may not be. Let's see what Visto says. Extinction is severe, but for introduced invasive species, we know the country did fine without them before. Yeah. And these two kept going back and forth. How many people get asymptomatic dengue infections where mosquitoes carry the virus? I think the number of asymptomatic dengue is very low. I don't know it offhand. Let's look it up. I can't type with my microphone here. Asymptomatic dengue infection. So now I have a crappy keyboard that's very quiet. And it's hard to type. Oh, look at this. I was totally wrong. 80% of all dengue infections are asymptomatic. So much for me. I admit I'm wrong. I got that wrong. So how many people? 80% are asymptomatic. And if you're already screaming, okay, you're not screaming. Good, but I admit that I'm wrong. Dr. Susan Oliver did a good job on RFK2. The word bollocks appeared prominently. More people need to do it, yeah. The RFK Jr. campaign had an unchanged MAGA legal notice on it, which they forgot to change, and the campaign had a photo selling hoodies using a Russian woman on a Russian street with Russian signs. Excellent. They won't get in trouble for it, right? Hey, Rima, I got a picture from someone today of their cat sleeping on a copy of my virology textbook. You said, everybody loves cats, right? So, yeah, a cat sleeping on the virology textbook. Isn't that cool? I should send it to you because I know you like cats. Welcome back. There's a cat in your little icon there. Yes, so your wholesaler sells this IPV with DTaP, right? So you can get it in combinations if you want to do that. If you want IPV alone, it's just Sanofi. But, yeah, the piggybacking is a good idea. You get multiple vaccines at once. But I don't think it's needed for IPV if you had it 25 years ago. It is a full-scale battle to control the narrative. Facts don't seem to matter. Yeah. Sam Harris of the Making Sense Pod is problematic because he platformed Alina Chen, but his last episode criticized Rogan and others platforming RFK Jr. Those audience crossover, so that was good. Yeah, Alina Chen, not good. No data on what, well, she says it came from a lab. There's no data from her on that. As a nurse attorney who worked for a healthcare system, we always hired experts in the science, medicine to review issues and prevent them. Yep, present them. Oh, Rima had COVID. Sorry about that. Symptoms like influenza. I did not lose smell. I'm glad you're back. Sorry about that. The Royal Society UK gave the David Attenborough Award 2022 for communicating COVID to Professor Jonathan Van Tam, Deputy Chief Medical Officer UK, did a lecture last week. Cool. Why have vaccines been such an issue with people compared to most medications or other medical interventions? I don't know. I don't think that other things have been issues also, right? You pick your intervention, people complain. People complained about molnupiravir for a long time. They complain about Paxlovid, blah, 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 rebound supposedly, which is not a thing. I think they pick on a lot, but as Steph says, I see down there, vaccines are required, right? And people bristle at being required to do anything. Yeah. This is a cowboy country. In many countries, they line up for their vaccines. The public health people say, take your vaccine. It's good for you. They say, where do I go? In this country, I want to choose what I put into my veins. And while I'm at it, I'm going to put a lot of alcohol in them as well. Makes perfect sense, right? Lee said, Les is here. Paul Offer writes books looking at pharma disasters like the Cutter incident. In his latest book, he's the last person to call a pharma shill. Yep. Misinformation in a lot of cases creates confusion, and confusion creates more converts for people who see things only in black and white. Misinformation is for recruiting, you bet. I heard it reported that the two lineage in the market is disputed as just poor samples with degraded sequences. That's not true. They're very good sequences, and so Holmes has said to me that that's nonsense. It's just sour grapes. Yep. See, I say Neumann's microphones are really sweet. Yep. Okay, Eric Topol saying tonight, new CDC data from blood donation showing at the end of 22, 1 in 4 are still no vids. Aren't some of these tests not helpful for vaccinated, though? At the end of 22, 1 in 4 are still no vids. Yeah, some of them, I don't know what tests they're using. They're probably looking at N protein antibodies, which is not 100% accurate, but I think it's good enough for these purposes. But I think that number is correct. 96% infected or vaccinated in the U.S. Never going to get anyone, although eventually everyone will get infected. New Zealand closed to visitors from China, but not Europe. And Italy was awash with the virus. By the time we closed completely, it was too late. Okay, got it. Remote audio gets messed up if you take it off the hall speakers. No, we didn't take it off the hall speakers. We took it right off the board, of course. We had an XLR out of the board. So we had wireless mics going to a board, and then the board out to a recorder. That's what it sounds. But then you get the room sound, because the sound is also coming out of the ceiling speakers, and it's causing a lot of echo and reverb. That's the problem. We don't go to the camera input. We go to a recorder next to the board, and then we sync it up in post. So we got it covered, Peter. Been doing this for years. Not to say I can't learn something all the time. Yeah. Heard on UCSF COVID grand rounds. Take it with a grain of salt. Mutation rate of SARS-CoV-2 is 4 times that of the flu virus. That's wrong. Oh, first of all, nucleic acids don't evolve at the same rate. So we have the fundamental mutation rate of the polymerase, which for RNA viruses is much higher than for DNA viruses, because DNA viruses have error correction mechanisms. The coronaviruses, however, have error correction mechanisms. The only RNA viruses that have such a thing. So their mutation rate, their basal mutation rate, is lower than that of other RNA viruses. But that's the basal mutation rate. What they're talking about here in grand rounds is not the mutation rate, which is defined as how many mistakes the polymerase makes every time it copies a genome. What they're saying is the emergence of variants. And that is, as Trevor Bedford said, is higher than for influenza virus. That's true. But I would not call it the mutation rate. It's the emergence of variants, which is a selection process on mutations that are generated. So the mutation rate of SARS-CoV-2 polymerase is lower than that of flu and other RNA viruses, but the emergence of variants is greater than that of flu. That's how it should have been stated. Grand rounds should not be making fundamental conclusions about virus biology. Do the Amy Edition shirts come in cashmere? They don't, but they should, shouldn't they? And that's with this lovely, there it is, the Amy Edition cashmere shirt there with the tails of the shirt sticking out. That's pretty cool. I like that. And Remus says, good quality merchandise. Thank you for purchasing. I totally agree. I didn't have facts. I took the J&J. It's just a bit annoying to hear people not using real facts to promote. I agree. I totally agree. It's very hard, as you see from tonight's discussion. The real facts are hard to get through out there, right? All right. This is the same question we addressed. Hopefully you understood it. It's not the mutation rate. It's the emergence of variants. That's higher for COVID-2 than flu. The basal mutation rate for SARS-CoV-2 is lower than that of flu because of error correction. Okay? Nothing changed. You shouldn't be calling it mutation rate because what you're seeing is the emergence of variants after selection, and the mutation happened earlier. I hope I explained that. Marco, maybe that's not for me. Ian, thank you for your contribution to science. It would take several orders of magnitude longer to do several orders of magnitude less than you do. Bravo, ragazzo. Thank you. Molte grazie. If the virus came from the market, how was it in the U.S. in December? It wasn't. It was in the U.S. in January, not before January. Okie dokie. What else do we... In Sweden, they say, if you talk about trolls, they will come. Looks like it is working here. Yes, they all started to come here. I saw that. Okay, so here's an example of someone who thinks they have a lab leak epidemic. Okay, Crystal, you should go listen to the TWIV we did at Penn State with Gigi Gronval, who has studied this particular outbreak of 1977 influenza. It was the result of a vaccine trial. That's not a lab leak, okay? Respectfully. They were putting it in people, and it spread. It wasn't supposed to spread. Not a lab leak. If you put it in people, you could get a pandemic, and that's what happened. So don't use that as an example of a virus that escaped the lab. They took it from freezers. They injected it into people, and it spread. So, nope. You know, I appreciate the respectfully. You're thinking, I don't know about this, right? And it's escaped my recognition. But no, it wasn't a lab leak. And then you're going to tell me, oh, the escape of anthrax in Sverdlovsk was a lab leak. No, it was a bioweapons lab. They're not doing anything to have any containment. It's not a laboratory where they're doing experiments. It's a bioweapons lab. Don't put it in the same category. All right. It looks like we have got to the end of your questions. I'll give you a minute to come up with some more. But in the meanwhile, I want to thank all the moderators. I'm going to get off early tonight, right? All the moderators tonight. We had Les, we had Andrew, we have Steph, we have Tom, we had Frank for a little bit. Anybody I'm missing? I said Steph, I said Les. Vanity Nutrition popped in. Thank you all for moderating once again. Really appreciate it. Peter, from Houseboard, so might be your mics. In my experience, might need more. Yeah, so the problem is, Peter, they gave us lavaliers. In my experience, having 4 or 5 open lavs in a big room sucks. No good. I would rather use the SM58's handhelds as really exclusion, and you can hold it close to your mouth. That's what we did in Houston. I think that's the solution. So that's what I'd like to do from now on. Yes, it's the open lavs in the room. Just they pick up the 2 Omni. Totally agree with that. Yeah. Noir, my daughter got me a TWIV tote bag. I love it. Wonderful. We had 200 people, 194 likes. Can't ask for more. That's great. Really appreciate it. All right, everybody. Oh, hello. Millie from China, Xiamen, China. I'm going to be visiting China in November. Going to be visiting a couple of different cities. What does John say? Just another comment. In TWIV 500, Vinny thought it might be 2028 when TWIV 1000 would occur. Little did we know. Yeah, we didn't know. Thanks, everybody, for stopping by. Next week, we'll be back. I'm going to see if I can convince someone to join me here. So it's not just me, although I understand that it's okay with you guys. But in the meantime, have a great week, folks. Next week, Wednesday, 8 p.m. Eastern, we will be back. Thanks, everybody. Good night. See you next time.